All right. Yeah, thank you, Tom, and the speakers of our first session. I'm excited, I guess I should say, to host for the second session today. We'll get a bit of mix of topics, including model validation methods, as well as kind of user perspective on some unique challenges associated with working with RNA molecules. We'll just get right to it. My pleasure to introduce first speaker today, Greg Pintilie from Stanford, who will be giving us a talk on 3D-EM map to model validation using Q-scores. Thanks, Greg. Take it away. We have you muted at the moment.
Yeah, it's always the classic mistake.
Right.
Okay, thank you very much for having me. It's been a pleasure watching all the presentations so far. So so we'll be following along, sort of along the main themes of map model building and assessment. Um, what I'd like to get into is, because this is about assessment, just to briefly review resolution, because that's a very common method used for assessing a map to begin with. And then I'll just show some examples just to make this kind of more comprehensive. I'll just quickly kind of show some examples of model building, a lot like what we've seen already. And then I'll get into sort of the main sort of theme of my presentation, which are sort of validation using different map model scores. I'll talk a lot about Q-score, which is something I've been working on along with many collaborators for the past few years. Show some examples and how it's getting implemented into the EMDB validation report. Okay, so to start with the resolution, so we have we build these maps, 3D maps from micrographs. Now, when you want to assess sort of the resolution of your map, what do you expect to see? We use this method where we split the particles into two sets, and we generate two half maps. And the idea behind this is that each half map will have will hopefully have the same signal, which is what we're looking for, but different noise. So this way, in Cryo-EM, as we know, noise is sort of the main thing to overcome. So we want to see to what degree the noise or the signal is above the noise. So we take these half maps, we turn them into Fourier space, and then we take incremental shells starting from the center, which represents very low frequencies, going outwards, which represent higher frequencies. And we see to what degree these shells correlate. And this gives us the FSC plot. And the part where the signal correlates well, we can trust the signal at these sort of spatial frequencies. But then eventually it will drop to some point where the signal is actually becoming overcome by the noise. And we get the resolution estimate. So in this case, let's say 2 angstroms. So what does this mean exactly? And this is in microscopy, resolution typically means, let's say you have a 2 angstrom resolution. That means you should be able to see objects that are 2 angstroms apart, let's say. So that means that, they have sort of well-defined peaks. They're usually represented by these smooth sort of peaks. But noise might, and this also kind of accounts for, okay, what we're calculating here is are these peaks distinguishable regardless of the noise that we still see in the maps. So what do we expect to see at different resolutions? So let's say something very loud just came on in the office. Okay, looks like it's dying now. So let's say we have a 13 angstrom map. Here we can sort of see individual proteins. So we might be able to build assemblies of proteins. At something like 6 or 7 angstrom resolution, we see secondary structures appear. So beta sheets like flat densities or alpha helices like tubes. At 3 angstrom resolutions, we start to see the pitch in the helix. We see separated beta strands, and we can even see some of the bulky side chains. And something like 2.6 angstrom resolution, now the side chains become better resolved. We might even see some ions, some blobs that represent ions. And in this case, this is a protein residue, and this is a ligand. So you can see a ligand can be very well defined at these resolutions. And then breaking the 2 angstrom sort of point, now the ions become more clear. You can even start to see some water molecules. And you'll see that the atom contours also become a little bit more defined. So you can start to build models with even more confidence than you might at lower resolutions. We've also been looking a lot at nucleic acids. So with nucleic acids, this is what a map at 10x resolution might look like, where you can sort of see the secondary structures. At 6x resolution, the ribbons of the become a little bit more defined. 3x resolution, you can start to see the bases a lot more clearly. You can even start to see ions again. 2 angstrom resolution, the bases again, and you start to see more atom contours, water molecules. And the highest resolution for RNA is a ribosome. And here you see the donut-like aromatic rings. So very, very, very good definition. Okay, now we've seen how we do model building. So a classic tool is Qt. So if you have density, that's pretty clear. You can start with, if you have some starting model, you can always sort of add more residues to it and interactively. There are some nice tools for building de novo models if the resolution is good enough. For example, Modelangelo, where it places some anchor points, it tries to connect the points, and then builds atomic models from that. Let's say you have a map that's slightly lower resolution, and we've seen some of this already. So here you take, you might have a structure that you already know, and you try to fit it into the map using, for example, exhaustive search. So some classic tools there are Citus. We've seen some other nice tools also in this workshop. Phoenix as a dock and map, and Chimera X, you can sort of do a semi-exhaustive search, or you can try to place this manually. And where it gets interesting is, let's say you fitted a model, but it doesn't maybe fit too well into your map. So then you can use something like refinement or molecular dynamic simulations to kind of jiggle it and hope that it eventually gets into the density. A very nice tool lately that I've been using a lot is Isolde for this kind of purpose. And you can sort of guide it by pulling and pushing on atoms as well. Okay, now let me talk a little bit about model. So getting into model assessment. So how do we assess a model? Okay, so as we all know, model has these atoms, and they're all the covalent bonded ones are connected with bonds. And here we usually measure a bond length or a bond angle. And from various high resolution structures, we've kind of sort of developed these statistical models of what an ideal bond length is, what an ideal bond angle is for different types of atoms. And then we have a equilibrium value and some standard deviation around that value. And we try to sort of, when we refine a model, we try to make them as close to the equilibrium value as possible. We also have these dihedrals. Excuse me just a sec.
We asked to talk to you.
Oops. It's getting a little bit noisy out the side. Okay, so with dihedrals, they involve 4 different atoms. So you can imagine it as rotation around the bond. And then we also have each atom might have some radius. So you don't want atoms that are too close together. So these are usually called clash scores. And then we have these special backbone dihedrals, which are characterized using these Ramachandran plots. So for a phi and a c angle, you can check if it fits within sort of previously observed values, which are known to be, which are thought to be energetically favorable. Anything outside is probably suspicious, and usually you, if it's not seen. Now, usually you don't refine. Well, technically, you shouldn't refine against backbone dihedrals. This should come out of these parameters mostly, but some tools will try to refine these as well. But this is also sort of a representation of model quality in a way, because again, it should sort of fit these previously sort of seen configurations. And then we also can look at rotamers. So each side chain can have a number of specific conformations that are known, again, to have been observed in high resolution structures or have good favorable bond angle dihedral parameters. But we use these rotamer libraries to kind of tell us, okay, is this side chain sort of reasonable? And then all of this considering that in some cases there might be possibly alternate conformation. So in some cryo maps, some side chains can be very well resolved, which means that this is a very sort of good fitting conformation. But in some cases, the side chain might disappear in the density, or it might actually sort of fork like this, and then you can even build sort of alternate conformers. Okay, so this is sort of like the kind of like the report I usually see for a model assessment. So very commonly used tool is the small probity assessment. So it tells you, okay, is the clash score good? Do you have a lot of rotamers that are maybe not favorable? Do you have Ramachandran outliers, bad bonds, bad angles, and things like this? So we use this to assess, how good the model is, like how good the structure is. And this is, yeah. And if you do go ahead and deposit a model into the EMDB, you will generally get a validation report, something that looks like this. And so far, it gives you sort of like an overview of some of these model-only metrics. So clash score, Ramachandran outlier, sidechain outlier. So this is good for evaluating a model. If you're depositing an RNA, they might also have, you also see something like this, which is the RNA backbone. And basically what these tell you is how good is this clash score relative to other structures. So in this case, the black bar is relative to all structures in the PDB, and the gray bar is relative to all EM structures. And one thing you'll notice that so far there's been no map model score that has been reported for this. And that's kind of something that's really been lacking so far. And that's something that that I've been looking at a lot. So to review this sort of map model scores were what we've been doing. So there's this atom inclusion, which is sort of like a classic score. Actually, the EMDB does give you an atom inclusion. What it basically entails is that you look at the atom positions and you use some contour level, which is usually defined by the user, and you say, okay, this contour level, is the atom inside the contour or is it outside? And if it's inside, that means it's good. If it's outside, it's bad. And you take this number that is inside and then divide it by the total number of atoms. And the higher the percentage, the better overall. I guess you could say the model is fitting into the density. With the main caveat that this contour level, if you give it a very low contour level, you might be including a lot of noise, and some atoms might appear inside where they're not. So something just slightly more sophisticated is you can take an average density. So you take the average density value at each atom position, and then you average it, divide by the number of atoms. And now this is not dependent on a contour level. So now the higher the average density, the better the model fit, let's say. But this only looks at atom positions. It's not looking at what's happening as you go away from the atom. Okay, and then finally, sort of like the sort of a more sophisticated version of this is, okay, we want to calculate a cross correlation. So here we take our model map and we generate a density map from this model. And in doing this, you have to specify a resolution. What resolution do you want this model map to appear to be at? So, for example, if you use a 2 angstrom resolution, as we saw before, you might see sort of well-defined atom contours. At something like 3 angstrom resolutions, the atom contours are not quite as well resolved. And then so to calculate cross correlation, we take the map values in your simulated map, and then you correlate them against the map values at the same position for each atom in the Cryo-EM map. So these cross-correlation scores will vary between -1 and 1. These are normalized. And then there's two different formulations for this cross-correlation. There's one where you subtract the mean, and then one where you don't. So this is like a very subtle difference. If you've looked at cross-correlation scores before, it's not always clear which one is being used, and there's strengths and weaknesses of both. Now, the main reason I think these never caught on and it's sort of used as a sort of validation tool is that it does involve this parameter that you have to specify. So So how do you know what parameter to specify, what resolution to specify? So, you can use the resolution of the map, let's say, right? But as we saw, that map resolution can be sort of approximate. Is it the right one? And then you might be tempted, okay, let me try to generate a map. It's lower resolution, one at higher resolution. Do I get it? And then you'll get a different score depending on what resolution you input into this. So it's very useful, if you want to see where your model is fitting the map, where it's not. But it's the main question that's always been around is what's a good cross correlation score? And you can sort of use this resolution parameter to kind of change it a little bit. So it was just very tough to use for kind of comparisons across maps and across different data sets. So what we started to do and this was kind of inspired really when these sort of atomic resolution maps started to appear, maybe 4 or 5 years ago, I guess now. This is a map. I think this might be no, there's a higher resolution map right now, but this is one of the highest resolution maps that we saw while we're doing this. Is that each atom is very well defined, right? So we started to wonder, okay, what does the density actually look like as you go away from the center of this atom towards as you go outwards? And what I forgot to mention here is when you generate these maps, usually you just place a single Gaussian at each atom position and you sum these Gaussians. Now, some more sophisticated ways of doing this is to use these scattering factor functions. And what these do is they will actually use multiple Gaussians, depending on sort of the properties of each atom. And it can get a little bit more fancy. But for the most part, if you just place a single Gaussian at the atom position, you get something that kind of looks like a Cryo-EM map. But here we started to kind of reverse sort of engineer this. Like instead of kind of, just placing a Gaussian, now we want to see what does the density profile actually look like in these maps. So we took map values at increasing radial distances away from each atom. And here we try to sort of focus on one atom at a time. So you'll notice that the points that we are considering are only the points on a sphere that are closest to that atom we're looking at and not to any other atom. So that's why these spheres kind of look incomplete. So these are the points that are closest to this atom at the top and not to any other atom in the model. So this way we can actually focus on each atom. It's not going to fully sort of ignore the effects of the scattering of this atom on this atom, but it tries to anyway. And okay, so not surprisingly, these atomic map profiles are Gaussian-like. So the purple curve is the actual experimental map values around this atom. And the black dotted line is an actual Gaussian function. It looks like this. It's just an exponential, which is actually fitted to this map profile. So we kind of least square fitted a Gaussian to this function, basically. And we can see that it fits reasonably well. Actually, if you look only at this point here, it fits to maybe like 1 or 2% sort of error value. So which kind of tells us, okay, a single Gaussian is actually not a bad approximation for the data that we see. It has this slight trailing edge, which kind of starts to increase, and this might be due to the effect of the environment of the atom. So as you go further away from the atom, 2 angstroms away, now other atom densities and things, solvent start to influence it, and it kind of goes up a little bit. So anyway, it wasn't surprising. It was single Gaussian, but I still remember this was a pretty exciting sort of thing to do and to discover, okay, a single Gaussian, one atom is a Gaussian. And then we did this with lower resolutions. So this was the 1.15 angstrom resolution. So a single Gaussian fitted to that. At 1.75, again, it's Gaussian-like, and we fit a Gaussian to it. And again, not surprisingly, it's Gaussian-like, and it has a higher sort of standard Gaussian width. The width is the sigma here in this equation. So the higher the sigma, the higher the width of the Gaussian. And as you go to lower resolutions, the Gaussian width increases. And that's how we would generate these maps usually. So if you want to generate a map at 2 angstrom resolution, you use a certain width. If you want to generate a map that's higher resolution, you use a smaller width. So we were thinking, okay, now how can we use this to assess, is an atom resolved or is it not resolved? And the first thing that I did was actually to try to fit Gaussians to the density, to the map values around each atom, but that wasn't too robust. Fitting Gaussians can, especially if the map, the atom profile is kind of flat, then fitting a Gaussian becomes sort of like sort of ill-posed in a way. So to actually come up to do a score, then to kind of come up with a sort of way of scoring or how well an atom is resolved, is we actually compared the map profile to a reference Gaussian. So we said, okay, what does a Gaussian look like at 1 angstrom resolution? And we roughly calculated that sigma, so it's sort of empirical. What is the sigma? It doesn't come from any scattering factors or anything. It's simply purely based on data. I looked at some high resolution maps, like these 1 angstrom maps, and I found out what the sigma was. And I said, okay, this is going to be the sigma of our reference Gaussian. And then we can take atom profiles at any atom and then compare it to this reference Gaussian. So in this 1 angstrom map, for example, for this atom, the atom profile is in blue. It looks a lot like the reference Gaussian. And we do a cross correlation. So it's still a cross correlation function. So the Q-score is basically a cross correlation between map values that you see in your map and values calculated using this reference Gaussian function. And you'll notice that there's these two parameters in the reference Gaussians, A and B. I won't get too much into, but these are kind of, we played around with what sort of values to use, and we came up with just with some sort of like a reasonable values calculated based on map values in your map. But really, the only parameter that goes into this Q score is this sigma. So for example, the sigma was determined based again on these highest resolution maps. So initially the sigma was 0.6, then when the highest resolution map was 1.5, and then when 1 angstrom maps came out, we changed this to 0.4 because that's kind of roughly the width that we saw in these maps. So we can do this for any atom in any map. In a 1.3 angstrom map, the atom profile starts to get a little bit wider, as we saw before. The Q-score gets a little bit lower. In a 1.7 angstrom map, the profile, again, sort of that we generally see is even wider, so the Q-score gets a little bit lower. And then on something like 2.3 angstrom map, again, yeah, the width gets larger and the Q-score gets lower. So what this Q-score actually does is it basically compares, your map values around your atom and your map and model to this reference Gaussian. And this fact that it got lower, based on sort of the resolution of the map, was sort of interesting. Not only because, okay, now we can evaluate, is the atom resolved or not? So if an atom is not resolved, as we see in some cases, for whatever reason, radiation damage or charge or whatnot, an atom like this might not be resolved. So the atom profile for this atom looks flat, and then you get a really low Q-score. So really, that's what we were after, is can we evaluate, at an atom level, is the atom resolved or not? So that was kind of what drove what the question that this Q-score resolves or aims to do. Now, if we take the average Q-score for all the atoms in a model and a map, and compare it to the reported resolution, as I've kind of been hinting before, the Q-score is, it gets lower as you go towards lower resolution. So we applied this to as many maps and models as we could. So we took about 10,000 maps and models in the EMDB, calculated average Q-score for all atoms in the model, and then plotted them against reported resolution. And just like we saw in those earlier images, the Q-score has this sort of relationship to the reported resolution. So the lower the resolution, the lower the Q-score, the higher the resolution, the higher this Q-score. And so it kind of what it sort of kind of affirms is that Q-score is measuring resolvability, much like resolution is. Resolution is meant to be a measure of resolvability, and the Q-score tells us how well atoms or groups of atoms, in this case, you can apply to any group size that you want, in this case, the entire model. And what was interesting is we saw this relationship. I don't have it written here, but the R squared, right? The correlation coefficient was something like 0.7, 0.8, which indicates a reasonable sort of correlation. For perfect maps and models, the relationship is actually the correlation sort of coefficient is 0.9. It can be very high. But the reason, is many points are actually sort of far away from this curve. So if we look at, okay, so if we look at the points along this curve, we see what I was showing before is that the lower the resolution, the Q-score kind of drops. At first, it drops in a linear fashion, and then we didn't initially look at this 5 to 10 angstrom range because the sigma of the reference Gaussian is actually pretty small. So so it starts to drop lower here, but still it stays above 0. So 6 angstrom map, you're still getting 0.2, 10 angstroms you might expect, or based on what we see in the EMDB, what sort of the expected value would be something like 0.1. And then what we did is we kind of went a bit further and developed a more of a statistical model of this, asking, okay, around this central curve, what are the 95 percentile bounds of the observed structures? And this is using statistics to kind of identify outliers. So if your point is within this 95% bounds, it's like, okay, maybe it's reasonable. If it's outside these bounds, there might be something is just not possibly right about this map and model. So we can look at some examples. So for example, number 1, we can see that the resolution is 2.36, but we get a Q-score of 0.7, right? So so there's clearly something off. And then if you look at the map and model, it's just shifted slightly. The map is very good, but after you originally fit the model, you get something that's more expected for the Q-score. In the case of number 2 was also interesting because this had a higher Q-score than you might expect to see at this resolution. And in this case, if you look at the map, it looks noisy and kind of broken up. So this looks like the map was likely over-sharpened. And then if you fit a model into an over-sharpened map, you might stretch all of those structural parameters. You might strain them a little bit more, and then get them to sort of get the atoms to move into more noisy sort of sharp peaks. So the Q-score does go up. But again, the fact that it's above this 95 percentile sort of range says, okay, you shouldn't be seeing Q-scores that are maybe this high. So you might want to check what it kind of indicates a slight issue possibly. In the case of number 3, another point here, which is actually the Q-score is 0. It's a 5.6 angstrom map. So it's outside of the 95 percentile. And here again, we see that it's just an incorrect fit. So after you rigidly fit it, you get a Q-score of 0.23, kind of what you expect to see. And then number 4, it's also sort of like another point where it's way above what you might has a Q-score that's way above what you might expect to see. And in this case, the reported resolution was 7 angstroms, and the Q-score was 0.36, which is again sort of high for this. And the reason for the higher Q-score is that a lot of the map actually is resolved reasonably well. Something maybe closer to a 4 angstrom resolution, right? If you look where this point might be, it's closer to 4 angstrom. But for some reason, the resolution was reported to be a 7. Okay, we can also look at per residue Q-scores. So here we generate a Q-score for atoms in the backbone and also in the side chains, and then we plot them on this graph. And here, why this is useful is you can sort of plot these 95 percentile lines as well. And then you can see, okay, which points are, above or below, and this can sort of guide to have a look there. Are these atoms resolved? They might not be resolved, or maybe in this case, maybe they're just not fitted properly, so you can try to fit them a little bit better. You can do this for proteins or for RNA. So this is a plot for Q-scores for here. We look at phosphate groups at the phosphate group, which is this phosphate, which is the backbone of the RNA. The sugars, which is the aromatic ring that connects to the phosphate, and then finally the base, which is on the end of the nucleotide. And here we can see that again. If you look at the Q-scores here, you can see regions that are not resolved, and then they have sort of Q-scores outside the range. And then there is residues that are well resolved, and those are within the range. So the reason this is useful is just as a sort of visual kind of tool. If you want to interpret your map and model, it can guide you to say, okay, this part of the model maybe I'm not so sure of. Maybe it's flexible. Whereas this part of the model where the Q-score is high, I can have higher confidence in the model. It can also be applied to other atom groups such as glycans. So here you can plot per glycan sort of Q-scores. And you can see that you get reasonable Q-scores when the glycan is resolved and poorer Q-scores when they're not resolved. And also something more interesting is when we look at ligands. So for example, in this map of beta-galactosidase, there is a ligand that's part of the model. And we actually had, as part of what this work came out of is a lot of sort of validation workshops or validation efforts that have been going on over the time. And this was actually a model ligand modeling challenge where we sort of challenged participants to build a model of a ligand or refine it. And what was interesting is actually two different models of this ligand were actually submitted to this challenge. So one, it's very subtle difference. This ring is pointing the same way, but this aromatic ring here is actually flipped 180 degrees in this versus this. And based on the density alone, they both seem reasonable. So you can see this O5, this oxygen atom here kind of fits into a density and has maybe some potential interaction. In this case, it sort of fits the bump a little bit better. So visually, both kind of seemed reasonable. If we look at the Q-score of this ligand was slightly lower, 0.6. And this was actually close to the 95 percentile sort of line. So this, I guess, could raise a bit of a flag that maybe your ligand is not fitted properly. Maybe you should check the conformation. For this conformation, the Q-score was more as expected. And then if we look at a map of the same, a map that's actually has the same reported resolution, but seems to have a bit more detail, then we can again check these two conformations, right? It's exactly the same system. It's exactly the same protein complex. So in this case, the Q-score, if you take this model and refine it into the map, the Q-score here still stays really low, so 0.5. So again, even more of a flag. And if you look at the density, some of these atom contours give it away that actually it's just not fitting that well. Whereas in this case, with this conformation, after refinement, everything, like all the atom contours match the bumps a lot better, and you get a Q-score that's again more in line with what you expect to see for this resolution. So I think in this case, sort of the takeaway is the Q-score can really sort of help you to assess, is my ligand well fitted? How confident am I in this in the ligand model? And I mean, ultimately, the higher the Q-score, the more resolved your ligand is, the more confidence you can have in it. So it is resolution dependent, and which makes sense. The higher the resolution, the more confident you can be in your ligand conformation. And lower resolution maps, you might get more into these sort of situations where like different conformations look reasonable, but it might be hard to tell which one is likely to be the right conformation. Okay, now back to just recently, maybe 2 or 3 years ago, I think the EMDB and the community kind of realized that there's no map model assessment score in the EMDB, and they started to sort of test integrating of Q-scores. Now, as I'm doing this, let me check if let me pause a little bit here to see if there's any questions or anything so far. Okay. Why shouldn't we refine dihedral angles? Going back, okay, this is an amazing question. So, and this has to do with validation. So dihedral angles, technically, it shouldn't be necessary to use them in your refinement because, again, the proper dihedral angles should come out of the other parameters, the bond angles, bond lengths, and bond dihedrals, which are well characterized for those atom types. And it was actually introduced as a validation score. Like are your dihedral angles sort of, kind of fall in these expected ranges that have been plotted before. Once you start to refine against the dihedral angles, then you start to strain your models to try to fit these. And the way that a protein model works is, you might strain other things. You might get higher clash scores, higher worse bonds. And it really was just a validation method to see if you've done the refinement well. And when you start to refine against that metric, then it becomes less useful as a validation metric. What's the easiest way to calculate Q-score for your map and model? Okay, I'll get into this. There are, okay, it sounds like somebody wants to chime in on this. Steve. Okay, maybe not. Yeah, I'll get into this more. There are two tools right now. The first implementation of Q-score was in Chimera. And now there's also a plugin for Chimera X. So those are the two main ways to do Q-scores. Two other libraries have come out that I saw, which I can point people to, but yeah, the primary ways right now is through Chimera and Chimera X. Okay. Let's see, let me manage this Q&A. Get back to the presentation. Oh, okay, this is still staying on. Okay, so yeah, so as I mentioned, the Q-scores are now starting to be reported in the EMDB because, you know, it's useful to kind of see where are the Q-scores low, where are they high, and this can sort of help to guide the interpretation and just sort of overall fit to map. Now, I guess one thing I've kind of one thing I said at the beginning is that the Q-scores are meant to measure the resolvability of an atom or a group of atoms. Now, this assumes that your atoms are actually fitted into the map. If your atom is not fitted into the map, obviously you're not going to have a Gaussian bump around it, and the Q-score is going to be low. Now, it's hard to separate those two. Is your resolution measures the signal in your map. And then I mentioned that Q-score is related to resolution. Now, Q-score is not directly meant to measure resolution. So because your Q-score is low, doesn't mean that the resolution of your map is low. It just means that the atom might not be fitted to the map as well. If it's fitted properly to the map, then it will be correlated to resolution roughly and resolvability, as I mentioned. So here, this is when you're looking at these, you know, Q-score sort of visualizations, you have to remember that it's a representation of resolvability of the map, but also possibly fit to map. And we'll talk more about this too. Now, one thing that is not in the EMDB that I've shown before in this presentation, which is actually, I think, kind of useful is, okay, for a given resolution, what is the expected Q-score? So that way, when you see a Q-score of 0.6, 0.5, or 0.6 or 0.7, what does this mean? Well, if it's close to that sort of expected resolution or expected Q-score, then you kind of know, okay, I've kind of fitted my model as well as I possibly could. So I think that's very useful, and this is not reported yet, but that's okay because really that's meant to be more a guide for you when you're doing the model building and assessment. And if I go back to these, all of these plots are basically made using the Q-score plugin in Chimera. So the Q-score plugin will calculate the Q-scores for every atom in the model. And then it will generate also a text file. This is all in the documentation. I'll have a link at the end, or you can search Q-score Chimera X. And then you can plot these Q-scores, you know, using some plotting program. I usually do it in Excel. So that's kind of the general. And then Chimera and Chimera X plugin will also have a visual representation of these. So you can sort of click on a point and go to there and then check the map and model yourself as well. But that's kind of what it's meant to be. It's sort of like a visual guide of sort of fit to map. The EMDB is also reporting the overall Q-score. So you'll see, as I mentioned before, the atom inclusion has been around. It's been reported for some time. Now the Q-score is reported along with it. So this is and it's doing the Q-score per chain. So here, you know, it kind of helps to see, is this chain resolved or not? Sometimes we fit entire protein complexes where some of the proteins might not be resolved. So you might see a low Q-score. And again, something that would be useful here is sort of, okay, what the expected or peak Q-score for this resolution is 0.5. So when you see your Q-score of 0.56, it's like, okay, this is, yeah, it's reasonable. Or it's kind of as expected. Okay, now in this latest paper that we did that describes a lot of the plots and the analysis that I've shown over the past few slides. We also came up with these two new measures. It's called a Q relative all and Q relative resolution. So what the Q relative all is meant to do is it tells you where does your Q score fall within all the Q scores seen in the EMDB. So for something, for example, for a map that's 1.9 angstrom resolution, is very high resolution. The Q score is in the top 1 percentile. So it's above 99% of all the other maps and models in the EMDB. And there's also a Q relative resolution, which is how far is it from sort of this expected or the commonly seen Q score. So, and the way that this is measured is we look at a bin. We bin sort of maps and models into resolution based on resolution. So we have bins like maybe, let's say, from 0.5 to 1 to 1.5. And then within each of these bins, what percentile is it within that bin? So this compares your map and model Q score to maps and models at similar resolutions. So this is something that's a bit more realistic. Let's say if you have a map that's 2.9 angstrom resolution, actually this is still pretty high. So this is it has a it's within the 71st percentile for compared to all. But it's actually kind of like low compared within its own bin. So it's only 50%. So let's look a bit more at each of these and what they represent. This is now in the paper, we only kind of printed this table. This visualization is not in the EMDB yet. This is kind of how I sort of envision it possibly looking. It's not really there yet. This is still kind of work in progress. But so let's say for this map, which is about 2 angstrom resolution, we can see the side chains are well resolved. So the Q relative resolution so the Q score compared to all maps and models in the EMDB, it's very high. Now within its bin, it's about 70%. So that's kind of reasonable. So within that bin of maybe close to 2 angstrom resolution, this is within the 70th percentile. So it's still very good. Now when you see a Q score of close to 0, this is likely a problem, kind of as I shown before. This is one of the examples I was showing before. But here, if you look at the map and model, it's slightly shifted. So yeah, Q scores are like Q relative all and Q relative resolution that are very low are likely to be problematic. And this, yeah, and for some reason, it was just hard to see this before when you're depositing your map and model. This map and model is still in the EMDB, but when people deposit it, you might not see, oh, I should maybe check the map and model fit. But hopefully with tools like this, it will become a bit more obvious. This is another map and model from the EMDB where the Q score is close to 0 and is just an incorrect fit. And this is a sort of, and this is the other example I showed before where the reported resolution is 7, but the map is actually higher resolution. So in this case, the Q relative all is maybe 30%, which, so it's within the 30th percentile for maps and models in the EMDB. With a Q score of 0.36, it's not high, but it's still above, it's but if you look at the Q relative resolution, it's actually really high. It's 100 percentile. So what this means is that this is high for the resolution of bin of 7 angstroms. This Q score is higher than every other score. So that's kind of also indicating of a slight problem. In this case, the problem is not really the map, it's just sort of the reported resolution, which was reported to be 7, is not really fully representative of the map. The map is overall, it does have these regions that are low resolution. So maybe this kind of biased this overall resolution report, but the most of the map and model is actually this higher sort of like is actually more resolved closer to, as we saw, like maybe 4 or 5 angstrom resolution. Let me pause again for a second. Let's see if there's any other questions. I'm not seeing I think it's because it's open. No.
Yeah, I can guide you through them. The Q score is calculated by Chimera X and the EMDB deposition site are apparently different. What is the cause for that?
It could have to do with the sigma factor. I'll have to double check. in Chimera X, it might be, and this was also something that came up with the earlier versions of the Chimera plugin, where initially we used a sigma of 0.6, but when EMDB started to integrate them, they used the sigma of 0.4, which allows us to compare maps and models at up to 1 angstrom resolution. So that's likely the right reason. I'll have to double check in the Chimera X plugin what sigma is being used, and I think it's adjustable. So if you want the same value, then make sure that, and maybe, you know, like I actually had prepared a slight small sort of demo. So let me just even check right now myself. So if you so that in Chimera X, we have this map model Q score. And then, yeah, that's, I think that's exactly the reason. So here you can see the reference sigma is 0.6. Now, it doesn't, it's not, you can use 0.6. You could technically use a reference sigma of whatever you want. But the ones we've used and we've done all of these statistical models on initially was 0.6, but now all the statistical models are based on 0.4. So if you want them, if you want to match what EMDB is doing, you just have to use 0.4. When you're looking at your own map and model, it doesn't matter what value you use because you're not interested in comparing across maps and models. It's only important to use the same value when you're comparing, different maps and models.
Does a selected binarization threshold affect the Q score?
No, there's no consideration of any threshold for Q scores. But related to that, because it's an interesting question, and I sort of hinted at this a bit, there are these two parameters A and B. So these parameters, you just want to make sure that your reference Gaussian is high enough. You know, it's not, the height is not 0, and the offset really doesn't matter because and this I tested and played with a lot initially. If we look at the formulation again, what we do when we calculate this correlation, we subtract the mean. So what that means is you might imagine that if you subtract the mean from this Gaussian, its center is going to go to 0. And then you subtract the mean from this atom profile, and it also, like, its center will always also go to 0. And it just turns out that this formulation where you subtract the mean is slightly more robust than this. And it's I'm not sure I can explain why, but I think when you subtract the mean, you know, you start to get negative values. And if you're multiplying negative values, but in your signal you have positive values, which let's say is like the complete opposite, it just makes the score a bit more, I guess it spreads out the score and makes it a little bit more robust. Yeah, but yeah, just slight little details that were kind of discussed a lot in these earlier papers, but it's always kind of fun to think back at them. Any more questions? Pressing.
That is it for now.
Okay. Okay, let me show so I mentioned that initially, and in the recent paper we put out, the statistical model was based on about 10,000 maps and models that were up to maybe about 2018. And now we've applied it to to most of the maps and models in the EMDB, which is about 26,000 so far. And I was curious to see if, you know, these relationships changed. If we look at sort of the peak, which is kind of the highest observed values for a given resolution of Q scores, it didn't change too much. Maybe it dropped a little bit. The low 95 percentile bar seems to have dropped a little bit more noticeably, which is a little bit worrisome, because what that means is that there's starting to get more points under this curve. So all of these points here outside of the curve, I haven't checked through every single one of them, but yeah, and I think what this kind of is telling us is that we do have to kind of have these metrics that help people to kind of you know, guide in model building and refinement, and help to find issues before deposition, hopefully. And yeah, so this is a summary. Kind of, we've looked at these Q scores per residue and visualized on the model. Now this has been kind of work that's been ongoing for many years, really, so far. And these are some of the advisors and collaborators that I've kind of driven it. Wachu at Stanford, Helen at PDB, Mike Schmidt, who was here as well, Kathy Lawson, Stephen Burley at the PDB. And these are sort of the new collaborators, Cal Morris at EMBL and Zhe Wang, who are integrating the Q-scores into, so I've been working with them a lot to do this integration and also to publish this latest paper in Acta Crystallographica. And this is a picture actually from one of the workshops that I mentioned. So this was all funded by the NIH over about 10 years. Basically what we did, the idea was to bring the community together and kind of discuss model refinement, model building, model assessment. So this kind of came out of all of these workshops and collaborations. And actually, I wanted to leave time. How am I doing for time?
You're good. You have like 7 minutes.
Okay. All right. I was hoping for more because what I wanted to do is I wanted to show you this Q-score plugin in Chimera X. But that might be enough time. Okay, so here what I did, I've kind of prepared this. This is a map. A lot of you will recognize this as apoferritin. I downloaded this map of apoferritin, and I did a quick sort of AlphaFold prediction. So the model is actually an AlphaFold model, and I took this model and fitted it into a low pass version of the map, because if you low pass it, it kind of helps to fit it a little bit more easier. So you can see, yeah, I think I have the right fit. And then if you look at the original map, it's a bit harder to see sort of the correspondence, but okay, let me show these and hide these. Okay, so you can see the fit is actually not too bad, but it's off. It's an AlphaFold model, so it's not going to fit the experimental map exactly, and that's kind of what I was going for. So as I mentioned, you can download this Q-score plugin. So it's, if you go to tools, you most likely won't have it initially. So you would go to more tools, and then you could go to, you can either search for Q-score, or there is I remember there is a model validation. So you can go here, and then it's already installed for, in my case. And I should mention this is not my creation. This is done by Tristan Kroll, who also did Isolde. My implementation, my implementation is the original Chimera implementation, but we're slightly migrating more and more of the functionality into Chimera X. But this was an excellent implementation of the Q-score by Tristan. And actually, this kind of takes me back a little bit, because it reminds me, Tristan actually, for the most part, replicated the method exactly, except for one little detail, placing points on those spheres that are increasing in radial distance. So to get back to the question whether the Q-score is going to be different than the EMDB, EMDB is using the Chimera implementation. This implementation might give you a slightly different Q-score as well, maybe within from my measurements, it was maybe like 1 or 2% error on average. So just because the implementation is different, but on the other hand, the implementation is a lot faster. So Tristan actually managed to make it a lot faster. Okay, so once it's installed, you go to tools, validation, map model Q-score. And so you'll see this dialog pop up. For me, it was docked here, but you pick your model, and then you pick your map, and then you click calculate, and you get this plot. Kind of like to have it docked here. Let me make the one a little bigger. Okay, so we see that the points are kind of all over the place. If I click on some of these points, it will take you exactly to that point. And you see that, yeah, the model is not fitted perfectly well yet. And this is actually where we can be adventurous and try to use Isolde. Now, if anybody has not used Isolde, you would install it also through Toolshed. Toolshed, you also have to install Clipper. But the first thing you do is you do Clipper, associate, and then you give the map number to the model, to number 3. Oh, yeah, that's right. 2. Okay, so now it's associated. You also have to make sure that there's hydrogens in the system. And then you can do Isolde start. I think it was already started for me, actually. So I can choose the model. Working on number 2. Okay. And then I want to go back because the whole point of this was to see how the Q-score plot can help in this process. So let me go back to here. Okay, so this is my initial Q-score plot. And then if I go back to Isolde and do the refinement, okay, so we see that everything kind of falls into place. Everything is fitting a lot better. And then I can pause the simulation, go back to this, and then calculate. And now, okay, now you see everything is a lot better. So the refinement is doing well, but you can still kind of click, you can still, so some of these low points, you know, you might want to look at them because refinement is not always perfect. Some side chains might be a bit too far from the density and you might have to kind of pull on them. Yeah, this is something I've always loved when Tristan was doing, is when you would focus on a residue. You start the simulation and then you kind of drag it into the density. Sometimes it takes a little bit more. Okay, now it's looking, this might be, yeah, you can sort of see how it's kind of going into 3 different directions. So this is possibly an alternate conformer. Oh, it went away again. Yeah, you can kind of see there's like 3 bumps here. So you can kind of pick which one you want to find. But once you've done this, okay, this was 166 I was looking at. So I think I was clicking on one of these points over here. Now you can see that point went all the way up there. So this is a very cool visual way of you know, checking your map to model fit using the Q-scores and this plugin and kind of helping to find issues. And also when you're interpreting the model. So if you click on some points, some of these residues, these glutamates, there's not much you can do. You're not going to be able to increase the Q-score by simulation in this case. So in that case, you know, it can help to guide interpretation. How confident are you in that model? Here's another residue, which seems to be outside. If I go to, this is residue 140. So that's one of these over here. So here, if I drag it into the density, you can see that went up. This one, that's the aspartate. Here's another aspartate. Oh, so this aspartate actually looks like it might have some density around it.
So Greg,
Yeah.
I'll say this has been a really cool demonstration of how users can incorporate Q-score kind of live with their model building.
Mhm.
In the last minute or two here, I might try to feed in some last couple questions from the audience.
Yeah.
A few of the questions, just try to kind of summarize a little bit. A few of them relate to I guess issues with refinement or incorporating other tools in refinement process after using Q-score. So one user asks, you know, after you do these adjustments, do you then go into Phoenix for refinement? And I might add to that to say, is there any development towards Phoenix using Q-score in guiding refinements as well?
Yeah, that would be great. And I've talked to some of the developers at Phoenix. I think they had sort of a tentative implementation. They asked me some questions about some of the details. I don't think it's officially released, but they are. Yeah, I think they did start to implement the same the algorithm. It's yeah, it's funny because it's sort of cross correlation is very simple to implement generally, but Q-score there was just this slight technicality of kind of looking at points that are close to one atom but not others, which kind of made it is kind of was the main bottleneck. And this was again to try to focus on one atom at a time. And this was the part that made it a little bit tricky to implement. So it's not the easiest implementation, perhaps. I'm not sure if they haven't released it because they had issues with it or something else. Yeah, that might. Yeah, we kind of we meet them at a lot at these workshops quite often. So, and so we kind of update each other a little bit. But we haven't worked too close. I haven't worked too closely with the Phoenix team in particular. I've used Phoenix a lot.
Yeah.
For refinement, things like that. Yeah.
Well, I want to respect the time for the next speaker, so you've done a great job kind of addressing questions throughout. There's a couple more if you have a minute to look at the Q&A.
Okay.
And we'll get on to the next speaker.
Thank you.
Thank you. It was a great talk. Thanks.